Here are the, if I can just pull over the slides here. We're still with this slide deck here. One thing that we're talking about is gene prediction. That means we're looking at sequences of DNA. Somebody brings you a sequence of DNA. You've never seen this before. You have no idea where it came from. And somebody says to you, I want you to annotate this thing. I want you to look inside this sequence of DNA and tell me where the genes are. Let's say I need to know. I need to know from my, maybe this sequence of DNA was extracted from somebody who has some variant of COVID, and I need to know, or maybe it's some other virus, but I need to know what the genes are that I'm looking at to make this person sick. And so there's a bunch of good reasons why you need to do that. And so I think one of the most important slides that we had on Wednesday was this one here. There's a bunch of slides we looked at here, but how do these prediction algorithms work? I mean, how do you know what a gene is inside your DNA? Where does that information actually come from? And you found that there's an alignment-based way of finding out like what the genes are, and by that we're basically using some kind of a local alignment. Remember what a local alignment is? You're looking at the actual subsequences inside a main sequence. And you're figuring out what what chunks of the subsequence have been found in other sequences somewhere else before. So in other words, you're looking at like a sentence and you're pulling out the words per words, and you're finding out what other sentences do I know where this word was found in terms of like the English language or any human language. So that was alignment-based. And then we have sequence-based, where we're looking for like actual start and stop codons. So sequence is like you're actually reading the DNA, and you're trying to use the knowledge that you have about the DNA about where genes would start and where they would stop. Now, typically this works. Typically when you find a start codon and then you find a stop codon later on, that means that whatever happens between is a gene. But it doesn't always mean that though, because remember these codons, stop and start codons, they're really just 3 bases, right? The start is 3 bases. That's that methionine. And then you have a stop, which is another 3 bases of DNA that you're looking at, or RNA if you're looking into RNA. But it's 3 bases nonetheless. And so you're saying that what happens between this 3 bases and what happens between this 3 bases is the gene. And while that's a good strategy, 3 bases, I mean, in evolutionary time, you could easily have some kind of a mutation, perhaps, where that mutation, where you have the gene has, or it looks like a gene is there, but actually it's just that particular part of the DNA you're looking at was actually a mutation. It's not real DNA in some senses, and so it's not really the signal that you're looking at to say that the gene exists in there. So that's sequence-based. Then you have the more commonly used, the probabilistic base, where you're looking at a whole bunch of different kinds of tests. You're looking at, for instance, well, again, you're looking at the alignment base. You're looking at global and local alignments of the subsequences inside your DNA, and then you're figuring out like what's the chances of what's the probability that this stuff would show up there randomly. What are the well, then you look at like other kinds of structures. You find, for instance, there's here are some other subsequences that act as what they call promoters, and you're saying, well, what are the chances that those particular types of regions would just randomly show up in my sequence? I mean, why would I have this thing here? Could this possibly be some kind of initiator for a gene? And so you have like these kind of sites, these words, if you will. I mean, I'll call them words. They actually do call them words in DNA, but they're like collections of like maybe 4 to 6 to 8 bases long, but they're actually kind of they're words in DNA. They're spelt in DNA bases. They're not words in human language, but they're words in DNA language. And so you'll have a combination of like 4 to 6 to 8 bases. I mean, it doesn't have to be even numbers all the time, but you'll have these areas which act as bonding or docking sites for specific types of biological machinery when the DNA is being worked on to create, when it's being translated or transcribed into RNA and then translated into protein. So you have these specific areas. And so you're asking yourself, well, what are the chances that these things actually just show up here randomly? And then you have like other types of content-based systems here where you're kind of looking at patterns, just other kinds of patterns like here. What are the frequencies? What are the words? What are the k-mers? What are the, I'm finding a whole bunch of these kinds of signatures could that represent, this gene? And so I would say that this whole slide here is probably kind of the summation of what we've been doing all week, just looking at these different ways of finding genes. Remember, all they do is they tell us that I believe that there is a gene right here at this specific place here. Anyhow, so we're looking at different types of these are all this like different these different systems of kind of analyzing genes to see what's inside them. And I think that we ended up. Well, actually, this is where things are getting interesting here. Frequencies of different types of words, and here I've actually worked in this area before. This was part of my research when I was in undergrad when I was doing my doctoral studies. That's like one of the first projects that I had, though, and that was analyzing again like the frequencies. This is like our frequencies, probabilities inside pieces of DNA to determine whether we're looking at a gene or whether we're looking at something else. And so here, this is actually a very interesting slide, another interesting slide. They're all interesting slides. But here we're saying that there's two different organisms, and the way that they spell out the sounds is different. So we have two different people here, and the way that they speak is different. Here we have two different organisms, and the way that they speak to their proteins, or they create their proteins, is different. And so you can use that kind of, these kinds of markers, if you will, here to determine whether something is a Homo sapien type of gene, if you will, or it's an E. coli. The only trouble with this is that E. coli is like the biggest mooch that there is. And across all organisms, I would say that the E. coli gene is the biggest mooch. And a mooch is one who basically borrows as much stuff from friends as possible. We're not even friends, but anybody. And so if you look at DNA from E. coli, you'll find there's a lot of stuff in there that came from other places. E. coli seems to just grab everything. And the reason why you might find E. coli, why there's like a lot of gene transfer going on with E. coli, and maybe some other many genes that might have originated here in E. coli, is because E. coli is so present. It's so everywhere. And so if you have E. coli pressed up against other bacteria, then you get the likeliness that you'll get horizontal gene transfer. But it lives in your gut. I mean, right now you have E. coli inside your gut that helps you to digest your food. I mean, you need that. That's part of your digestive system. But you have viruses that have taken out pieces of your code, who infected you, carried that code somewhere else, maybe got trapped, or maybe there's an E. coli somehow that got in the way, that interrupted the virus, and the virus exploded, and its DNA was floating around, and then E. coli picked it up. Who knows? But there are a lot of overlaps. But we're really looking at, for instance, we're looking at the commonalities between these types of different organisms and how the amino acids are used. But one of the interesting things I think that we ended up here is we were talking about these boundaries, and the boundaries are actually quite difficult if you're looking at a gene, you find that there are intron and exon areas. The intron areas are these non-expressed regions, and the exons expressed are expressed regions. And so if you're looking at a gene, for instance, if you think that you found a gene and you're looking to see whether this is actually a gene, you're looking to see whether you have expressed, non-expressed, expressed, non-expressed. You're looking to see whether you have these regions inside that gene. And so you're really looking to see whether, can you find boundaries? And the problem with boundaries is that they're not conserved, and so it's difficult to have a specific one specific signature for a boundary. So this is interesting. And so what you're doing is you're looking to see whether the code has some kind of, you're looking at the code to see whether it has some kind of change or some kind of change, or maybe a signature or something that you recognize as you go through this coding, non-coding, coding, non-coding, coding, non-coding region inside your gene. So you have what they call this sliding window approach, which is actually quite clever. But what it is that if you're writing a computer program that does this work, you would have your DNA, and you would have a window that kind of defines a start location and an end location. And so you only analyze those particular bases in your DNA in that window. So in this case, the window is 5 bases long, and so what that means is that your window is going to be looking only at 5 bases. So you'll be looking at, for instance, the top window here, which is 64337, and those values represent different types of measurements that you might be, the frequency, for instance, perhaps of those codons, something like that. There could be other things too. There could be the energy. Every codon, for instance, every amino acid has a specific type of energy that is a physiochemical property. And so maybe you're sliding through your proteins and you're looking through those kinds of energies to see whether you've seen those signatures before. But this sliding window is very common in bioinformatics. And then what you do is for this first window, you record a score, you put that down in your list. And then for the second window, you're just sliding it over by one. And then that becomes the second item on your list. And so there's a lot of overlap between your windows. You'll notice that, for instance, the first window is 64337. Those are my data points that I'm looking at. And then next, I still have the 4337, but now I have 2 when I slide the window. And so there's a lot of overlap in data between these 2 windows. But actually that's okay though, because when you, just FYI here, when you get around to plotting this data, if you have the 23 and 27, if you look at the sequence of DNA and you plot 23 and 27 and then 21, and you keep on going for the duration of this whole sequence, it's what they call a smooth plot, which, if you remember from your mathematical days in calculus, your smooth plots are going to be kind of like more gradual and less spiky and rough and wild and crazy. And so you can actually make more kind of, you can actually see kind of better, a more global view of what's going on in terms of the energy of these, or the energy scores of your amino acids. The reason why I would say that is because if I just had a window where the first window was 64337, and then the second window began with 2, 1, 5, and so on and so forth. In other words, I have 2 windows where there's no overlap. Then my graph or my plot is going to be very jagged and aggressive looking. But here, though, when there's all this overlap, it becomes a smooth curve, and that in some cases allows you to actually see the essence of what's going on. You can actually see the trends, which you wouldn't see otherwise. That took me to work that out. I worked that out. I wrote 2 different programs where I used a sliding window approach for my analysis, and then I used another kind of sliding window where there is no overlap whatsoever. And I was using the no overlap whatsoever approach first. When I was looking at the energy, the physiochemical, the energy of proteins. And I just couldn't see any signals that were coming through, and that was because the windows, there was no overlap, everything was bouncing all over the place. It was like a lightning bolt through my data. It's like I couldn't see anything. But when I began like having this overlap of the sliding window where only one base differs from, or actually I guess only 2 bases differ from window to window, then it became more gradual and you could start seeing trends showing up, which was actually quite a lot of research at the time. I was so glad to find that. I went up to this guy in the hallway, and I'm like, oh my gosh, high five. And he's like, why? What is it? And I got my research to work. And they're like, cool. Like, yeah, man, glory days, glory days. Anyway, so we're talking about these boundaries. And another thing that we can do, though, for instance, is you can look at what's called the Codon bias index, where you're looking to see, in a nutshell, you're looking to see how the codons are used. As we discussed before, this is related to this page here, and that is that different organisms prefer using different codons to create their proteins or to code for amino acids. So for instance, here last time we discussed that an E. coli prefers UUU to create that particular protein or that particular amino acid, whereas in UUU over UUC, everyone see that over here, these 2 ones on the top? But if you are human, then it would appear that UUC is a better codon for that particular amino acid than UUU. And so different organisms have different preferences for how they like to build their proteins. I guess that makes sense because different children have different favorite bricks and Lego that they like to use to build things. And so why not? Why can't organisms have different amino acid building blocks to build their proteins? So going back down to this, you're looking at those types of codon biases that exist between these exon and intron boundaries. So supposing I know, for instance, that there's a particular organism out there, and they like to use this particular amino acid in their in their genes. If I know if that's a fact, and I'll have to find that fact by other research, but if I know that that's a fact, then I might be able to use that inference to say, yes, actually, this appears to be a different kind of signal between the the coding region of my gene and the non-coding region of my gene. In other words, when I know that this organism likes to use UUU for its amino acid, when it makes that amino acid, and not the other, not UUC. And so if I find, for instance, in a region of gene, I find a whole bunch of UUU amino acids being, or UUU codons being called for, that would be maybe an exon, because I know that that organism prefers to do that. So it's a lot of detective work, and it's very exciting work too. I mean, it's like you actually are solving, you're actually solving a kind of a whodunit mystery here, because you have no idea really, what the codons are until you start kind of looking around and kind of getting an idea about what's the neighborhood of this codon, what kind of what kind of selection or what kind of selectivity is there for that codon. But you're really just looking at what's called the frequencies of these codons. You're just trying to find out, like, well, I mean, it's just a frequency after all. It's just a frequency. But you're looking to see. You're saying, here's a sequence, and I'm finding a lot of these codons that are showing up in the sequence. I know that this organism likes using those codons. It's a common codon. They could have used something else. I'm going to guess that this frequency, this high frequency, is indicative of a coding region inside a gene. Another thing that's actually kind of interesting, and when you see this, it's like, it just blows your hair back. It's like your eyes are wide open, your hair is being blown back. It's very cool. But they have these islands that exist inside genes. Or these, actually not necessarily genes, but they're the islands in other places too. But they're islands that are called the CPG islands. And what these things do is they are between these exons. They are boundaries still, I think. But these CPG islands are like, you'll find a region of code where everything's kind of like, there's not an unnatural amount of C and G combinations or pairs in your DNA. That's what CPG, C paired with G. But you find a whole bunch. I mean, you'll find in your sequence, you'll find that there's a region down here which doesn't have a whole bunch of C's followed by a C followed by a G, and then suddenly you do. And this is kind of like my window, my sliding window here. And so this is what I meant, kind of like by it's kind of a smooth area, but I'm making a sliding window, and I'm just counting how many times I see C's followed by G's, not G's followed by C's, but C G pairs. And so if I find C, G, C, G, that counts for 2 pairs. And I'm not looking at the C or the GC pair. That doesn't. I'm just looking at how many times I find a C and then I find a G. And so you'll find that there are these regions inside your code where suddenly you have a whole bunch of these things. Suddenly you have a whole bunch of C, G pairs. You can hear that bang, bang, bang. And that's the monster in the basement, kids, so listen up. You better eat those Brussels sprouts, otherwise he's going to come up and get mad. I'm just kidding. I would never say that. That's mean. But you can hear the bang on the pipes. Anyway, but still we have this. So we have these regions like that, and then suddenly those regions disappear almost as fast as they pick up, and that is that suddenly everything goes down to this. Like there's this area where there's like nothing. There's no more trend like that. So it's like it's low, and then it goes up, and then it's low again, and it's just as sudden as that. And if you find something like that, if you find what's called a CP island, where in the midst of your sequence, you suddenly have this area where you have lots of lots of C's and G's, CPG island rather. If you find an area like that, it could be the indication that you are between your intron and exon regions. Like in other words, you found a border. And so this is kind of interesting here. This is just an analysis that I found on, I think, Wikipedia, which is actually kind of interesting. And the reason why we care about this is because these types of areas tend to be found, I guess, just before the gene would begin. So, for instance, when a gene is about to start, you'll have a whole bunch of these CPG sites and sometimes the GPC, in other words, CG pairs or GC pairs. But you'll tend to find a whole bunch of those things kind of accumulating before, in this case, before genes would start. And so maybe, and there's some theories about this, but maybe you have, these are pieces of, I remember G's and C's have more bonds than A's and T's, and so maybe these are pieces of, or these are bonds that have been added to code, because if you have all this biological machinery to a piece of DNA, maybe that might rip it, might break it. You might find that there's a problem. And so maybe you have to strengthen the place upstream of the gene, in other words, like before the gene begins, where you go upstream to downstream of the gene. So you start upstream, then you move downstream of the gene. So upstream, before the gene begins, maybe all this biological machinery to copy the DNA and then to make RNA and then to do all this other stuff, maybe all that stuff has to stick to the DNA sequence, and maybe if you didn't have so many bonds in there, it might actually rip or break the DNA. I mean, it makes sense. If you have a whole bunch of people holding on to one line, maybe that line would break. And so there's a whole bunch of different theories. In fact, every time you look at the literature on this, you'll find that the theories become even more abundant, which is actually kind of interesting here. But they're saying here, though, that C paired with G's are more abundant in upstream regions of the gene where they form a CPG island. In other words, if you start finding this kind of motion here, this CPG island, you might think that following all this island, like here, I might expect to find a gene showing up somewhere. I mean, that's another signal you might find. Or it could be that these are between your boundaries, I guess, between your intron and exon areas of your gene itself. And so you have to kind of have an idea about what you're looking for. You know that there's a signal, but you have to be able to interpret that. And so this goes back to an original idea, and that is that you, as the bioinformatician, you have to use, of course, you have to use your analysis, and you have to be able to find this kind of phenomenon and be able to kind of and see it like this. But also, though, that doesn't get you very far, though. You have to actually use your intuition and your intelligence and your education to interpret what that could possibly mean. So supposing I do find this, does that necessarily mean that I've found an island, that I'm about to find a gene, or maybe I'm passing between boundaries? Does that mean anything? Maybe, maybe not. Maybe this is all random. But then if it is random, then you have to look at it in terms of like, well, could it really have happened by itself? I mean, could it really? Is there enough information to suggest that this really wasn't random? And so there you are. So that's why you have to like you have to kind of spend more time kind of looking at the data and trying to figure out like, does this seem to tell the story that I'm trying to find? Is that what have I found evidence for this whole thing? So there you have it. There's actually kind of an interesting thing. This is, by the way, in this particular data here, in the you may have noticed that in the dark blue, which is like right here, dark blue, that actually indicates the beginning of a gene, ATG, and then you find this TGA at the very end, which is the end. So we have the start and the stop. And so one of the problems you can see here is that you have a bunch of these islands before this ATG, but then you have, or maybe you can say this is the same island. I don't know, it depends on how you look at how you decide this. But you might say that in this island, let's say this whole thing is an island, inside this island we have this start area. And so maybe if you find an island, that means you have to kind of, if you find this, maybe you have to go inside that and see whether there's more information to give you an idea about if that actually is a gene, because you'd be looking for some kind of a start area. Okay, so that's actually kind of interesting here. But what I'm saying is that even though the data or your results may say one thing, it doesn't mean that that's necessarily the case. This is another picture here. This is using these C-paired G island or CG islands to find more areas which could be promoters, which basically means it could be the onset of a gene. The gene could be beginning around here because I found this unusual combination, or this high amount of G's or C's followed by G's. Not G's followed by C's, you don't count them. Of course you see them in the sequence, but I'm actually looking at C and then a G. So if I find a C and then followed by a G, if I find that pair, then that's significant in this case here. That's island. But if I look at G followed by C, then your results are different. And so it's not necessarily the same thing. And so you're looking for a C upstream of the G, in other words, if that makes any sense. Finally, when you find these kinds of islands, by the way, this is just like kind of give you an idea about what's going on here. I found this in, where did I find this? I found this online, and there's a paper out there. I don't think I put the source in. I can put the source in later. But if you find these islands, though, this is a paper really saying that in this case, this is one of those papers that's kind of arguing, it's arguing for a lot of things, but this paper is arguing for the extra strength that you get from C and G bonds when there are a whole bunch of them in your sequence, and that is that you have all these particular pieces here that you have all these, like, these pieces that will have to do some kind of work on the sequence, and when you have all those pieces kind of sticking onto the sequence, then they add weight and they start doing things and adding stress to your sequence. And so you need to have a bunch of these, what they call the CG islands, available so that they can support the weight and all the pulling around and the yanking caused by all this machinery that has to work on the sequence. And so they're saying, I mean, this paper here actually does say a lot of things. I can find the link for it, or the reference for it. But it's saying a lot of things here, but it's saying that because there's all this work happening on this particular piece of sequence that they were studying, this group, you have to have strength there to make sure that it doesn't snap apart. I mean, you can imagine how that would just, I mean, a piece of DNA is not very strong at all if you're tiny and you have all these pieces pulling and pushing and tugging and working on it. So it could easily snap. Yeah, that's basically what I wanted to say here. And so it's like you can, by finding these C and G areas, it suggests that something is going to happen there. It suggests that that's a construction zone of your DNA. Are there any questions at all over any of this stuff here? I wanted to, so we're done with these slides now. I wanted to go now into the next thing, which is an activity where I have given you a piece of code, and I wanted just to show you how to use this code. But the activity is here. You have until Monday to get this whole thing done. And what we're looking at now is we're using open reading frame tools. Now, when I was talking before about gene discovery or gene prediction, you're actually using, you're actually trying to find out whether there is some kind of are there embedded genes in a piece of DNA. And so they call those open reading frame tools. And the idea behind that is that you're looking at the DNA and you're trying to find out if there's anything in that DNA that looks like it could code for protein. But the reason for the frame, why do they use the word frame, is because in this particular case, in this picture, the graphic here, you can see that there is actually 3 different reading frames. There's reading frame 1, or sorry, reading frame 3, 5, and I can't see the one here. So I didn't capture the name. But basically, you have 3 different reading frames. And so that means that by changing the way that you're reading your codons, if you read at position 0 and you read position 0, 1, and 2, then that's my first codon that I'm starting with. I might get the first reading frame. But if I start with reading frame 1, 2, and 3, or rather base 1, 2, and 3, then I get a different selection of codons. We've discussed this. You get this reading frame, the second one here. Or if I start at reading frame, or if I start at base 2, and I go with 2, 3, and 4, going 3 by 3 by 3, starting at 2, then I get another reading frame. And so really, inside a piece of DNA, you might get several different proteins, which really are found in relation to where you start looking at your DNA to read the codons. So that's why they call them reading frames. But you're still running the same analysis, though, as we were discussing before, to see whether there's any kind of there's these islands, to see whether there's any probability, to see whether we've seen specific types of genes before that show up in that sequence. And so what I'm asking you to do is to work with a piece of code that I'm going to give you, that actually I have given you. It's in the source directory of your repository there. At least it should be there. Is it there? It should be there. If it isn't there, then I'll have to put it there, but I tried to make sure that it was there. The reason why I'm saying this is because I sometimes forget these things. I'll just go ahead and pull this over just to make sure that everything is there. But what I'm saying, though, is it should be there, and it's called the orf_finder. It's a Python program, and I wanted to spend some time just to talk about it, but I want you to go through these questions and remember that everything that's in blue is a question that I want you to respond to. Otherwise, everything that's in black is part of the steps that I want you to follow. And so what you're going to be doing is you're going to be using your BioPython Docker container. Please use that to run your Python code that you'll get in your repository. So that means you may have to, if this is a new install of your computer or something else, if something's happened to your old container, you may have to rebuild it. So I can show you in a moment how to do that in case you've forgotten. But you have to use that container to run your code. And the reason for that is because there is Python or BioPython in there, and it's also Python. So in case you don't have Python or BioPython, the container will allow you to it installs that for you and allows you to run that. The questions you're going to be responding to today are on this Google form, and I think that's basically about it. But you're going to be running this Python program, which is really going to be looking at doing the same kind of work as this. It's going to be making up its own assessments, and then I want you to use the NCBI Open Reading Frame Finder tool and run the same analysis. And I want you to compare and contrast what you find. And I think that you'll be, shocked at how different some of your results may appear. And I think that is something that you really must be aware of when you're using tools. I think that once you run the experiments, you'll see what I'm talking about, though. But I think that you'll be shocked at how different some of the results are. And when you think about how bioinformaticians don't always use the same kinds of tools to make an assessment, they'll just say, oh, I did this result, or I did this result, and I found this, this, and this. Well, while that's fine, it doesn't necessarily mean that that's the way it always is. Right? So it's like it doesn't always, just because you found those results in this particular case doesn't mean that that's kind of how it always is supposed to. It doesn't mean that that's the end of the story. That's what I'm trying to say. Let me just go ahead and get into my code here, and I'll just show you how to run the Docker container, just so that you don't forget how that works. Hopefully I can find this here. I'm just trying to get into there we go. Now I need to show you my terminal. If I can find it. There we go. Here, you should be able to see it now. Yes. Right, so remember where I am now. I'm going to be running, I have to run I'm inside here. I have my LaTeX, or actually not LaTeX, it's, you don't have LaTeX, I have that LaTeX, but you have your source. Now inside your code here, this is the piece of code that you're going to be running here, this ORF Finder Python program. This FLEC ORCID FASTA, typically your data files, if they're FASTA files, FLEC ORCID.FASTA, or FASTA. But I put this .txt at the end so that you should be able to go ahead and click on this thing with your computer and see the data showing up. I always find that if my machine's working with a .fasta file, it doesn't know what the file is, and so it doesn't know what to do with it. But you're going to be editing this file or this Python program, and so I'm giving you this backup directory here too, so that you don't have to go back and re-clone your ClassDocs repository to pull out the file again. But this is just a backup of the file. It's exactly the same. There's nothing different. I've just copied it there. And so when you're in one of the program steps, it asks you to make a modification to this code. If something terrible happens where everything is completely destroyed, where you can't run the program and you're getting frustrated, well, don't freak at all. Just rename this file into something else and don't remove it. Hold on to it until the end of the course. But go to your backup directory here, and then copy this file here back here and rename it as this. This file you would have copied someplace else and made it like .backup or something. So anyhow, I'm running this from my container, and so how I'm going to do this is kind of crazy here, but what I'm going to do, I think I have all my stuff here. Let's see, I have my Docker bio, and inside here I think I have to go to source, is that correct? And then I have to go to build. Actually, do I have? That's not that. Actually, it's here. This is using Mac. If you're using Windows, it may be a little bit different. I think your slashes are the other way around. But what I'm going to do, though, is that, notice, for instance, that where I am is, let me just go ahead and copy this, so I don't have to paste it again. But where I'm located, there's no Docker files. And so this is something I wanted to show you some time ago, but I just never got around to it. But what I'm going to do is I know that there are Docker files I need to use, which is all this stuff here. These are the files that actually come with my ClassDocs, okay? And so I'm using my Mac here, so I need to use this build, and I need to use this run right here. But notice these are not in my local directory. And so I'm going to use some directory maneuver here, which is actually kind of cool. I'm going to run that file. So I can use sh in my Mac to run the file. Back on the tree where I am, back again, back again, back again. Find this directory here. And by the way, you can always push the tab button, and that will give you kind of a view of what files are there where you are. So in other words, I'm staying put where I am now, but I'm actually kind of going into other directories and looking at the files. I'm going to go ahead and hit my build Mac, and if I can't use build here. I don't have my Docker file here. I think I just run this. I'll just run this. And there we go. And if I just type in this command here, sh // docker_bio run, then I can get into my container. And I know that I'm in my container because I have my root, which is right there. Hopefully everyone's getting there. If not, then there's other ways of doing this. But the reason why I wanted to do this is because when I type in ls, you'll notice that the files, because I stayed put in this directory, the files that I ran, or the files that were in my local directory where when I ran this command here to actually open up my container, those files now show up inside my local directory inside my container. So I can run these things. Now let me just show you something else here. I can go ahead and just run this file like this. There's online help, and so I have all this. But if you are not in your container, let me show you something else that you get. And that is if I run this directory here, if I run this in the outside, here I am outside of my container. I just left my container. If I run this file here, it will say, hey, wait a minute, it doesn't look like you're inside your container because I don't see any BioPython files showing up. And so that's why you'd use something like this to get inside your container. And again, if you type in this, whoops, you can run everything. So how you actually run this is you just type in Python 3, followed by the file name, or you just put dot or /. Goodness gracious, my fingers just won't work today. And this is how you run the program here with your parameter. Hopefully you can all see this. It's kind of squashed away at the bottom of the screen. Maybe I can move this up a bit just to kind of make things easier. I don't know if that makes a difference. If I make the screen a bit darker, perhaps that would. How do I do that? I don't know how to do that. Well, I think maybe that's good. Okay, anyway, but still, when I run this thing, you get some information that comes out. So that is all you get. I'm going to, I'll leave it there. And so now you know how to run the program. You know how to get into your Docker container. If you have any questions, you can look to each other. You can look to me. You can ask questions on Slack. But I think, though, that you have the makings of some capability in your program here. You'll see that it's capable of doing some pretty amazing stuff. But I want you to think about the capabilities of your program in terms of the capabilities of using NCBI's tool, which we found. Remember, how do I get back to this thing again? Goodness gracious, here. So when we go back to that's not it. It's right here. When you go back to your activity, it will ask you to go to. Oops, that's your thing. It will ask you to go to this NCBI. There we go. Orf finder, right here, and then run basically the same analysis all over again. Anyway, I think I've said enough. Please don't forget that you're using this Google form here, activity 10, April 23rd, and you have this some questions to follow. Should be done. Are there any questions? Oops. If there are any questions, I mean, if there's no questions here, I think I might end class now then. Of course, I'll still be available by Slack, and so if you have any questions, I mean, let me know. But I'll leave this to you to work on. Otherwise, that's really all I've got for today. This is due on the 26th of April, Monday. So if you have any, I mean, if you have questions, you've got time to ask. And by the way, if you do, you can ask them to me directly if you want. But also, I mean, put out a message on Slack in the general channel, and find out whether anyone's had that same deal or that same problem, and see whether they have any advice you could use. I mean, really though, I mean, work with each other. That's kind of one of the nice things about Slack is that if you have a question about something, to send it out. Hey, has anyone had the same trouble that I have? How did you fix that? And blah blah blah. And you might find that someone has just finished solving that, and they'd be more than happy to tell you exactly how they did that, which is so nice. Anyway, that's all I've got. I will talk to everybody on Monday. Everybody have a good weekend. Get some air. It's beautiful outside. Just beautiful. Just gorgeous.